Hey everyone, I believe we can go ahead and get started with this afternoon's event. So I will begin by first of all extending a warm welcome and thank you to everyone who has attended the first of its kind ANPA COVID-19 conference. So again, welcome. My name is Dr. Susan Anyanwoke and I am the membership committee chair. And I will just take a few minutes to share a very important message on behalf of ANPA. First of all, I would like everybody who, is maybe not an ANPA member and those who may be an ANPA member to remember that ANPA has a very rich history of philanthropy and advocacy here in the US and in Nigeria. It's been in existence for over 25 years, has over 20 chapters across the United States. ANPA, by all rights, is an extremely formidable organization. And we're an organization that is committed to evolution and modernization and hearing what our members want. We're committed to providing benefits, quality benefits for our members, such as the programming that we have today and our routine webinars that provide key financial information to our members. So I say this all to say what our ask is. And our ask from those of you who may not be members is we want your support. We want you to join us. We want you to hear what we have to say. For those who are members, we want you to help us to continue to grow. Again, help us to support our growth, not just through your dues, which are very important, but through your voice and through your time. Engage with us on all of our platforms. We have e-groups, we have WhatsApp groups. Most importantly, we have local chapters that will benefit from your time, your commitment, your voice, and your money. So, ANPA really meets a very important need for the Nigerian physician community, the Nigerian health practitioner community, and we need you to believe in us and to be a part of it and to help spread the word. We already know that there is strength in numbers and our collective voices really takes us very far. In this last final moment before I hand over this baton, I do want to remind everyone as the year comes to a close that the beginning of next year is renewal season. So if you have already not put your mind on renewing your membership dues, please do so. You will be getting some regular communications from Letitia, from the membership team, as well as the IT team on new processes for membership payment. From all of that, I say again, welcome. At this time, I would like to proceed by introducing one of the co-moderators for this afternoon's event. He is a board-certified, fellowship-trained orthopedic surgeon. He is a member of the Georgetown MedStar Orthopedic Institute in Washington, D.C. He is the co-founder of the Center for Advanced Specialty Surgery in Nigeria. He is an avid philanthropist. He's the founder of Operation Stand, Walk, Run, as well as the 2019 Humanitarian Award winner provided by HCA. And on top of all of that, he is the chair of the ANPA DMV chapter. He is a rather impressive and formidable ANPA member. I introduce to you Dr. Aham Onyike.
Good afternoon, everybody. Thanks for the very, very flattering introduction, Susan. It is indeed a pleasure to be a participant and a moderator for today's very important discussion on the COVID-19 vaccine. Once again, I'm the chapter chair of Washington, D.C., so I say compliments of the season from the nation's capital. The nation's capital, as we recognize, is the epicenter of COVID. It's the home of the U.S. Congress, which right now, as we speak, is meeting to decide on a $940 billion or possibly up to $1.4 trillion stimulus plan. Here in Washington, D.C., we just had the FDA yesterday approve the Moderna vaccine, which has equal clinical trial record with the currently distributed Pfizer vaccine. Here we have the NIH, which is the forefront of the research and scientific information pertaining to the vaccine. We have speakers from all over the United States that are outside of Washington, D.C., but connected to each other and have emerged in 2020 as the thought leaders authorities in infectious disease, particularly with COVID-19, and we're proud to have them as fellow Nigerians lead the discussion. So it's an opportunity to piggyback on what Susan just said, that ANPA represents the potential strength in numbers, and it's been on display more so than ever in 2020 with Zoom and other online platforms. And today, I'm very confident that we would all agree after the discussion that we do have the expertise within our ranks. We're going to have a broad and deep conversation on the COVID-19 vaccine, which has been led by many fellow Nigerians, some of whom are here today. In particular, I want to introduce Dr. Gertrude Anyanwoke, fondly known as Gertie. She is the chair of the, she's the medical director of the Infectious Disease Associates of Baton Rouge, also medical director of the Multi-Specialty Clinical Care Center, also in Baton Rouge. Gertie, in addition to that, is the chapter chair of the ANPA Louisiana chapter, the current committee chair of the Infectious Disease Committee for ANPA, as well as the chair of the COVID-19 task force. Gertie, as you all know, has been very visible, very prominent, very available in leading discussions pertaining to COVID-19 this year, and of which the hottest topic currently is the vaccine. So without further ado, I want to introduce an authority in COVID-19 discussions and the vaccine, Dr. Gertrude Anyanwoke. Thank you.
Thank you so much, Aham. That was really flattering. Without further ado, I would launch in to introduce our current Association of Nigerian Physicians in the Americas, ANPA president, no other person than Professor Chris Okunseri. Professor Okunseri is a professor of dentistry at Marquette University. Before we introduce the panelists, we would like to call on Professor Okunseri to give us a brief talk about ANPA. Professor Okunseri, you have the floor, sir.
Thanks, Gertie, and welcome everybody to this great opportunity, and especially thanks to the panelists. But I think another sincere thanks has to go to the Infectious Disease Subcommittee and the COVID task force that Dr. Gertrude Anyanwoke chairs. Gertie and that group have been working round the clock from the very minute we identified COVID to now that we have a vaccine. But I also would take this fine opportunity of our great organization to echo all the introductory remarks that have been made so far. Dr. Susan Edianwe reminded us of our great organization and why it's important for us to continue to remain members of this group, talked about our dues, talked about value of being a member of this group. But especially, I think this opportunity showcases all the collective strengths, talents, resources of our great organization. We can see the content. We are going to hear the content from these talents we have and the expertise of the panelists about the vaccine that would help all of us to actually try as much as possible to bring this pandemic under control. So our organization continues to be great. We are very honestly grateful for our membership, without which we would not be where we are today. And I encourage each and every one of us to continue to demonstrate and keep that flag flying. We are all proud of the talents we have in this organization, and we will continue to use our collective strengths to make sure that we achieve our vision of a healthier Nigeria in a healthier world. So on that note, I want to say Merry Christmas to all of us and our families, and ask that we all continue to stay safe as we listen to all the experts this afternoon sharing content and experience with us. Thank you very much, Dr. Anyanwoke.
Thank you. Aham.
Thank you, Dr. Okunseri. current ANPA president. Also want to acknowledge immediate past president who is here, Dr. Shemaine Emelife. Our next introduction will be Dr. Adedeji Adefuye, popularly known as Deji. He is one of our panelists for today's dialogue. He's currently the Executive Medical Director of Takeda, I hope I'm saying that right, Pharmaceuticals, and he's an immediate past chair of the ANPA Infectious Disease Subcommittee. He has been very involved in the pharmaceutical industry in a lot of the progressive recent developments, again, pertaining to COVID-19. And he is going to bring that vantage point to today's discussion. Dr. Adefuye, you're welcome. We will introduce the other panelists as well, and then our main speaker will go ahead and give a lecture to the group. After that, then we'll have the panelists participate and lead us in discussions and hopefully have a very interactive session where we can answer all the questions. They invite the tough questions as well. So Dr. Anyanwoke will introduce the second panelist.
Thank you, Dr. Aham. I have the pleasure of introducing Professor Igho Ofotokun. He's a career clinician scientist, and his work, a lot of it has been centered on people living with AIDS. His resume is very long. I will not be able to read it all up because it will take all our time today. But Professor Igho, in a shortened form, is a professor of medicine at Emory University School of Medicine. He's a member of the FDA Antimicrobial Advisory Committee. He's a member of the IDSA governing boards and the HIV-MA governing board. He's a member of the NIH Infectious Disease Clinical Research Consortium Leadership Group, which is in charge of coordinating the whole COVID-19 vaccines, not just one, but all of them, and the therapeutics. Without further ado, we welcome you, Professor Igho Ofotokun.
Thank you. And then I'll introduce our speaker.
Thank you. Thank you very much.
Okay, you're welcome. So again, the format is that our speaker, Dr. Obi Nnedu, whom I'm about to introduce, will get things going by giving us a lecture on the vaccine. So as a way of introduction, Dr. Obi Nnedu is a fellow of the Infectious Disease Society of America. He's an infectious disease expert. He was trained in internal medicine at Tulane University, followed by a fellowship at the University of Washington. He's currently a Center for Tropical Medicine Program Director, Infectious Disease Fellowship at Ochsner Clinic in New Orleans. He's an expert of the Coronavirus Prevention Network and clinical investigator doing research in the COVID-19 vaccines. And so he brings that expertise to the ANPA community today. He's been a thought leader and an authority in his community and also nationally, and has his fingertips on the pulse of everything related to the COVID-19 vaccine. Without further ado, Dr. Obi Nnedu, the floor is yours. I believe our technical crew will let you share your screen and then you can go ahead with your lecture.
Absolutely, thank you very much. I am going to share my screen. All right, can everyone see my screen?
Yep.
And everyone can hear. Good. And everyone can hear me okay?
Yes.
All right, so I'm going to go ahead and get started. I was asked to give this talk, and I'm very, very happy to give this talk to everyone. I was also asked to try to be somewhat brief, just so that we can have time for discussion. So if I'm going through slides a little bit quickly, just stop me and ask questions or type in the chat box. I'll try to keep an eye on that. As far as financial disclosures, I don't have any. I just wanted to show this map just to let everyone know kind of the gravity of things and where the hot spots are. I'm in Louisiana and New Orleans. New Orleans was one of the first cities that was hit really hard. Shortly after our annual Mardi Gras parades, we were really slammed. I lost a lot of my patients in the process. And what you can see is that fortunately, Louisiana is not as bad as other parts of the US. This is from the CDC, and I just got this yesterday. I think everyone on this call is familiar with the fact that there's been a number of cases, and unfortunately over 300,000 deaths, some of which are healthcare providers, just like we are. So very somber numbers, and more reason why we need to double down on our public health efforts, and why we're cautiously excited about the EUA approvals for these COVID vaccines. I want to touch base a little bit, just show a rough cartoon of the structure of the SARS-CoV virus, and specifically talking about this spike protein here that allows the virus bind to its target cell, get internalized by the target cell, and ultimately cause some pathology. This spike protein has been the target of essentially all the vaccines, and part of the reason being that it's relatively conserved across different strains of the SARS coronavirus. And the other part being that studies have shown that antibodies to that spike protein are neutralizing, meaning that they prevent the virus from doing what it wants to do. So we're optimistic. We know that people who get infected seem to develop some immunity. There have been very, very few cases of confirmed reinfections. And so most people seem to get some type of immunity. We don't know exactly how long that immunity lasts. As I mentioned, the spike protein is relatively conserved across different strains of the coronavirus. And so what that means is that if you create a vaccine and you create immune responses to that protein, it in theory will still be effective against multiple different strains of the COVID-19 virus. The current vaccine candidates being studied in the US, there are about 6 of them, all showed promise in preclinical non-human primate model studies. And we have phase 1 and 2 trial data on most of them that were generally quite encouraging. We talked a little bit about the fact that the spike protein of the SARS-CoV-2 virus is relatively well conserved, which makes it an excellent target for vaccine development. So stages of vaccine development. One thing that our patients will keep asking us, and you hear this all the time, is, well, this was really rushed. They cut corners. I don't trust President Trump, and things along those sorts of lines. And I think it's important to state that these vaccines have gone through the basic stages of development, the exploratory stage, the preclinical stage. That's when you're testing the vaccine in non-human models, typically primate models, and these vaccines went through that stage. They're currently going through the clinical development stage, which will be your phase 1, phase 2, phase 3 trials. 2 of them have gone through regulatory review by the FDA and have been given emergency use authorizations, and obviously being manufactured and quality control. And so the vaccines have gone through the basic stages of vaccine development. Corners were not cut. Things were just sort of expedited because of the emergency that we're dealing with. So for the clinical development part, as I talked about, your phase 1 trials will typically contain just a small amount of people, and you're mainly just assessing the safety. You want to make sure it's safe in humans. The phase 2, you have a little bit more people, and you're assessing safety as well as immunogenicity. You want to make sure it's safe. You also want to make sure it's stimulating the immune system in the way that you want it to. Then with the phase 3 trials, that's when you're assessing the efficacy as well as also safety as well. and these are usually larger numbers. And so what a lot of the current vaccine candidates for COVID-19 did was they sort of combined phase 1 and 2 and basically assessed safety and immunogenicity with the smaller numbers, and confirmed that those candidates were safe and that they stimulated an appropriate immune response in the individuals that got the vaccines. So again, to be clear, they essentially followed the basic steps of development, and so nothing is no corners are being cut. Let me put it that way. So I just wanted to kind of just quickly talk a little bit about vaccine platforms. The live viruses, we all got measles, mumps, rubella. That's the actual virus that's been sort of weakened or attenuated in a way that stimulates your immune system without getting you sick. Recombinant viral vector, that's when you have an unrelated virus and the gene of interest is inserted in that virus, and that way when it's injected in you, you produce the proteins from that gene that prime the immune system and get you safe from that particular infection. A good example is the new Shingrix vaccine that's out there. Inactivated viruses will be basically a virus that's killed, and like the flu shot, protein subunits, that's an actual protein from the pathogen. A good example is hepatitis B. You mix the protein with an adjuvant, something that helps kind of stimulate the immune system a little bit extra, and then you inject it in the person, and you end up with a good immune response. And then the mRNA vaccines, these are relatively new, but have been around at least going back to 1990. And what's happened over time is that the technology in terms of storage and transportation of these has developed to where these are now viable vaccine options. So the qualities of an effective vaccine, you really want it to provide long-lasting immunity ideally. You want the vaccine to have really high efficacy. You want it to be safe, limited side effects. The effectiveness needs to definitely outweigh whatever side effects may come from it. And ideally, it should be easy to store and transport. And I'll just add cost effective also. So these are the 6 that have been in different stages of development here in the US. There is a Russian vaccine that apparently has showing relatively good efficacy in preliminary studies. That one is a recombinant viral vector vaccine, and it's certainly shown a lot of promise. But these are the ones that we have under study here in the US, one by Pfizer and Moderna. Both of those are mRNA. Recombinant viral vector vaccines are AstraZeneca and Janssen. And then protein subunit vaccines will be the Novavax, and then the time, I'm not going to go through too much about Sanofi, Novavax. Briefly, the Sanofi is apparently on hold. I think they're still kind of reanalyzing data. Novavax is supposed to be going into phase 3 trials this month, if it hasn't already. It should be going in sometime. Janssen, AstraZeneca, Moderna, and Pfizer, those are all in phase 3 trials already. And we do have some preliminary data from phase 3 trials from AstraZeneca, Moderna, and Pfizer, and I'm going to go through those shortly. So this is the AstraZeneca. This is a viral vector vaccine, and the viral vector here is the adenovirus. And what they did was give a dose of 5 times 10 to the 10 viral particles. These were 2 doses spaced out by 28 days. So this is a phase 1 to 2 trial. So really, they were in this particular trial, they're focusing on safety as well as, is it immunogenic? Does it actually prime the immune system? And so it was a 2-dose strategy, 2 doses spaced out by 28 days. And the vaccine was found to be relatively safe. And then in terms of immune responses, so this is the placebo, this is the meningitis vaccine. This is people who just got 1 dose. These are folks who got the full 2 doses. And then this arm over here will be convalescent plasma. So this is people who actually were naturally infected, and then you measure the antibody levels a couple of weeks, 2 to 3 weeks after the infection. What you can see is with the prime boost strategy, you're getting antibody levels that are basically on par with those who were naturally infected. So what this meant was, the data from this phase 1/2 trial essentially showed that you're getting good immune responses, comparable immune responses to someone who's naturally infected, and it is in fact relatively safe. So based on this, this particular vaccine candidate went on to phase 3 clinical trials, and these were recently published. And this is preliminary data from this particular trial. And so some of you may have heard of these results already. So at least at the time of publication, this many individuals had been enrolled. And the particular analysis focused on about 11,000 individuals that were enrolled in the United Kingdom and Brazil. And they got the 2 doses spaced out by 28 days. And so I summarized this table a bit on my next slide. But I just wanted to kind of say that overall vaccine efficacy was about 70.4%, which is good. If you consider that the flu vaccine in its best flu season would be somewhere between 40 to 50%. And so this is good. But what was interesting about this was there was an error in a small cohort of in this phase 3 trial. Some people got half the dose the first time, and then the standard dose the second time. So LD is low dose, standard dose will be the second time. And for some reason, those who got half the dose the first time and the regular dose the second time, in that subset, the vaccine efficacy was 90%. And then in the subset that got standard doses both times, the vaccine efficacy was lower. And so there's been a lot of discussion about this, and the scientists are certainly looking into that, and specifically looking into how the protocol was altered accidentally, if you will. But overall, the data is very promising for this AstraZeneca vaccine. Just to kind of summarize, those who accidentally got half the dose for the first dose and then the full dose the second time 4 weeks later had a vaccine efficacy of about 90%. Those who got standard dose twice, spaced out by 4 weeks, it was lower. And the overall vaccine efficacy was about 70%. There were 3 cases of transverse myelitis, one that led to a brief halt of the trial while they tried to investigate whether or not it was related to the actual vaccine. To my knowledge, it's not too clear, but it was deemed okay to continue the trial. The other 2 cases were deemed to not be related to the vaccine. And so based on everything, I think it's very promising, this particular vaccine candidate, in terms of its efficacy and safety. And a big plus about it is the issue of storage. It can be kept between 2 to 8 degrees Celsius for up to 6 months, which makes it something very promising, particularly for developing countries that may not have the ability to establish the cold chain for the mRNA vaccines that we'll talk about in a little bit. It also is being priced cheaper than the mRNA vaccines that Pfizer and Moderna have. So a little bit about mRNA vaccines. I think there's a lot of anxiety about it. Again, it's been around for a little while. It's just that we've made advances now in terms of how to store and deliver these. It's not infectious. It generally doesn't replicate in the person. Essentially, it gets into the cell. It uses the cell's machinery to produce the protein of interest. That protein is then presented on the cell's surface, and then it stimulates the immune system. Meantime, the actual mRNA particle is destroyed by the cell. You know, it has a half-life, and it essentially goes away, and you have the protein presented on the surface of the cell, and that sort of is what the immune system sees. It recognizes it as foreign, it builds an immune response, and then that immune response ultimately leads to some memory, hopefully in the long term, in the case of these COVID-19 vaccines. So there are 2 mRNA vaccines in trials here in the US. The Pfizer one, my institution is involved with, and then the Moderna vaccine is the other one. Talk just a little bit about the Pfizer one. I just wanted to show data from its phase 1/2 trials. Again, this is early clinical trials focusing primarily on safety and its ability to prime the immune system. Let's get through that. And so smaller numbers of people enrolled, a few different dosing strategies were used: 10 micrograms day 1 and day 21, 30 micrograms day 1 and day 21, and then 100 micrograms as a single dose. And this is just showing that this is immunogenic in terms of the immune response. So HCS is convalescent sera. So these are people who actually had COVID-19, recovered, and then you measure antibodies afterwards. And what you can see is that vaccine, gives you antibody levels that are comparable, if not higher, than those who were actually naturally infected. And that's really what you want the vaccine to do. The idea of herd immunity, from letting the virus run wild and killing people off, is, it would be almost unheard of in the infectious disease community. You want to achieve herd immunity through vaccines, and certainly this Pfizer mRNA vaccine appears to at least give a robust immune response and appeared to be safe in these early phase 1 and 2 trials. So based on that, the phase 3 trial went ahead. This was recently published in the New England Journal of Medicine. Again, this is preliminary data. This is data basically acquired 1 week after they got their second dose of vaccine. So still very early, 1 to 2 weeks after they got their second dose of vaccine. So still kind of very short follow-up for these individuals. About 41,000 people had received 2 doses. They used the 30 micrograms concentration and gave doses 3 weeks apart. And they assessed the efficacy and the safety in this phase 3 trial. And I just wanted to point out that there were about 8 cases in the group that actually got the vaccine. That's a little over 18,000 in this group that was analyzed, and about 162 cases in the group that got the placebo, and this led to vaccine efficacy of about 95%. And so we were extremely excited about this in terms of at least its short-term efficacy. These individuals will ultimately be followed up to 2 years to assess long-term efficacy in these individuals. This is just a summary of what I just went over. Again, 95% vaccine efficacy, very, very promising early data. They kind of looked at what they defined as severe COVID, and they were told of 10 cases. 9 were in the placebo group, 1 was in the vaccine group. So the vaccine, again, these are very small numbers, but there's a suggestion that it may protect against severe COVID. And then obviously there's this issue of storage. The Pfizer mRNA vaccine needs to be stored at -70 degrees Celsius. Once it's taken out, it can certainly thaw and stay at room temperature for up to 4 hours prior to use. Just some side effects. The most common was pain at the injection site. That was the most common local side effect, and probably the most common overall side effect that was reported by these individuals. Some of the systemic side effects were headaches, fatigue, muscle pain, but in general, these all resolved within 1 to 2 days after getting the vaccine. So that was the Pfizer mRNA vaccine, and I'm just gonna touch on the Moderna. First, going over data from the early phase 1/2 clinical trial. Again, this is also an mRNA vaccine. They used a 2-dose strategy with different concentrations in this early trial. Again, looking to see which would be safe and which would stimulate a good immune response. These injections were given 4 weeks apart, as opposed to 3 weeks apart, which was the case in the Pfizer trial. And I just wanted to show that they did, in fact, stimulate fairly good immune responses. All the different dosing strategies, 25, 100, 250 micrograms. The decision was ultimately made to proceed utilizing the 100 microgram dose with 2 doses spaced out by 4 weeks. You can see this last bar, this is measuring antibody levels. This last bar is convalescent sera. So these are people who are naturally infected, and then you measure antibodies 2 to 3 weeks afterwards. And you can see that your antibody responses are comparable, if not actually higher than those who are naturally infected. And so based on this and the fact that in the phase 1/2 trials, this appeared safe, it went on to phase 3 trials. The phase 3, the early preliminary data from the Moderna phase 3 trial, to my knowledge, hasn't been published in a scientific journal just yet, but it has been released, early data has been released to the press. I just want to say that these preliminary analyses are being generally done by a DSMB. So this is an independent body kind of analyzing the data. So it's not the actual company. I think that's another source of concern that many people have. And in this case, the Moderna vaccine showed a vaccine efficacy of about 94.5% in people who've been followed for at least 1 to 2 weeks after they got their second dose of vaccine. And so again, this is very high vaccine efficacy, very, very promising information. There were 11 severe cases of COVID that were recorded in this cohort, and none of these cases were in the vaccine group. They were all in the group that got placebo. So again, very small numbers, but certainly a suggestion that the Moderna vaccine also may protect against severe COVID. These are some of the side effects that were reported. Infections like headaches. In terms of transportation, this vaccine also needs to be stored at cold temperatures at -20 degrees Celsius. It's a little bit more forgiving because it can remain stable in a standard fridge at 2 to 8 degrees Celsius for up to 30 days. And then it can remain at room temperature after you take it out for up to 12 hours. So this is just sort of a summary. We have our 2 mRNA vaccines that we're excited about because they've shown some efficacy, as well as, certainly the efficacy far outweighs the side effects that have been reported in a small subset of people in these trials. They've both now been giving emergency use authorization by the FDA. The AstraZeneca vaccine also has very promising early data. Janssen is currently in phase 3 trials, and these are the 2 are a little bit earlier on in their development. I just want to end with myself getting a shot. I was one of the first people at my institution to get vaccinated. And that is my talk. Thank you guys so much.
Thank you so much, Dr. Obi Nnedu. That was very good and very in time. Dr. Aham Onike and I will be looking in the chat to see when people have questions, and we can answer some of them in the chat group. There are others that when we feel the whole group can benefit from it, we'll let the panelists answer them. So please post your questions, and also people can raise their hands for further discussions.